This is the University of Otago.
Good evening. On behalf of the University of Otago, I would like to warmly welcome you to this inaugural professorial lecture. I would like to extend the welcome to both the staff and the students of the University of Otago, and also members of the general public who so religiously attend these inaugural professorial lectures as part of their ongoing continuing education. As a group, we are all collectively here to support Julian because we realize what a significant achievement it is to be promoted to the rank of professor at the University of Otago. Julian's research and teaching here at Otago is actually a very cogent reminder to all of us that the work that we do here is not only important, but it is also useful. And like some of the work that we will hear about tonight, Julian's work actually addresses some of the major challenges that we will face in the 21st century. Long before the current emphasis on green energy became very, very popular, scientists have long been on a quest to mimic the way in which plants convert sunlight into fuel. In leaves, as I think we will probably hear tonight, photosynthesis converts carbon dioxide and water into oxygen and carbohydrates such as glucose. And the remarkable thing about this process is that the energy transfer efficiency is well over 95%. So the production of hydrogen using nothing but water and sunlight offers the possibility of an abundant, renewable, green source of energy for the future. Now, one of the major hurdles to solve in the mystery of how plants manage to break apart molecules while not simultaneously breaking themselves apart has been one of the rate-limiting steps in our ability to use the processes of photosynthesis and apply them to renewable green energy. And this is actually the mystery that some of Julian Eaton-Rye's research intends to solve. So today, we have the great pleasure of learning more about his groundbreaking research on Photosystem II. And I would now like to hand over to Professor Frank Griffin, who's going to tell us a little bit about Julian's journey to professor.
Thank you. Kia ora. Distinguished academics and friends. I've written down details here because many of the words are very big, and I will either forget them or mispronounce them, so I will stick to a disciplined invitation to Julian to give his inaugural lecture. It's my privilege this evening to introduce Julian Eaton-Rye and to provide some background information that's sometimes not even found in CVs, to obviously see the road he's traveled to become professor at Otago. Julian's early academic journey didn't always transverse the conventional academic pathways, so it's worthy of note. And I think it also provides a beacon of hope for parents. It began with him completing his secondary school CSE subjects in the UK some 40 years ago. He then spent some time as an auxiliary nurse in a disabled serviceman's home before damaging his back lifting one of the patients. This provoked him to continue his education and culminated in completing his secondary qualifications in 1977. He then carried out basic training in the army with the unfulfilled prospect of becoming a physiotherapist. Having been frustrated in that prospect, he decided to circumnavigate the medical school and go to the Department of Botany, where he in fact carried out his undergraduate studies from 1978. And he graduated with honours BSc Botany in 1981. And during that period, he also had annual visits as a camp leader in the United States. And this obviously made a deep impression on him, because having completed his BSc in Manchester, he decided to immediately go to Illinois to carry out his PhD studies, which he carried out and completed a PhD in botany at the University of Illinois in 1987. Two-year postgraduate fellowships were then held in Japan, and they were followed by two-year periods as a researcher and senior researcher at Arizona State University and Brookhaven National Laboratory, New York. His postdoctoral experience allowed something important to happen. It allowed him to transition from plants to the really important living cells, the microorganisms. And he was introduced to cyanobacteria, the fathers of all biological systems, and mothers. And obviously, these are very important organisms because oxygenic cyanobacteria are pivotal to life as we know it. For without them, we would be no more than undifferentiated slime wallowing in an anoxic primeval swamp, rather than being professors here at Otago. So we have a lot to thank cyanobacteria for. Julian was appointed lecturer in the Department of Biochemistry in 1964, and since then he's progressed senior lecturer, associate professor, to currently professor in sort of quinquennial movements. And he has been especially involved in sustaining and develop the area of plant biotechnology here at Otago. He has trained numerous postgraduate students, many of whom as graduates have become academics here in New Zealand or overseas. And as a testament to his skills as a mentor and supervisor, he has been given the OUSA Award for Excellence in Postgraduate Supervision. His contribution to his own field was recognized by the Outstanding Plant Physiologist Award by the New Zealand Society of Plant Physiology in 2005. He has been past president of the New Zealand Society of Plant Physiologists and the New Zealand Institute of Chemists. He currently is a member of executive international committees on phototrophic prokaryotes and the Society for Photosynthesis Research. He is deputy head of the Department of Biochemistry and a member of the academic board of the Division of Sciences and the University Board of Graduate Studies. Like all good academics, he has extensive international linkages. In recent times, he has given invited presentations at international conferences held in the Czech Republic, India, Thailand, Japan twice, Canada, Australia, Azerbaijan twice, Portugal, and New Zealand. It is my great pleasure to introduce Professor Julian Eaton-Rye to present his inaugural professorial lecture that no doubt will enlighten us about the pivotal role photosynthesis plays in a healthy, oxygen-rich planet. Welcome, Julian, a card-carrying botanist, a practicing biochemist, and a closet microbiologist.
So I'm going to begin by introducing us to photosynthesis. And in fact, the talk is really going to be about Photosystem II in photosynthesis, and it's going to be a fairly general talk. I'm not going to get into, I hope, details that are too nitpicky. And I hope that this is an introduction to a protein that I've worked on for about, 30 years. And it's meant a lot to me to do that and be in this field, and I hope that I can convey some of that to you. But before we find Photosystem II, I'm going to start with this picture of a tree and remind everyone that chlorophyll is the pigment that absorbs the energy from the sun. And as the Vice-Chancellor said, to begin with, we need to fix carbon dioxide, and that happens in the canopy of the tree here. And the product is carbohydrate. This is a C3 sugar called glyceraldehyde 3-phosphate. And the exhaust gas of the whole process is to evolve the oxygen that we breathe, which comes from the splitting of water. And this whole process relies on the energy from the sun, which is about 5,500 zettajoules, which is 12,000 times the annual global demand. So there's a lot of energy coming in. And so in a time when we worry about whether or not we have enough energy, we actually have the energy. We need the technology to tap into it. So in more ordinary terms, more energy strikes the Earth in 1 hour than humanity needs in a year. The scale of photosynthesis is immense. The green here and the blue are both actually positive indicators of photosynthesis in the oceans. There's more in the green area than in the blue, and the green areas on land, of course, are carbon fixing going on by photosynthesis, and the numbers are staggering. So the terrestrial organisms in the tropical forests and savannas fix around 85 times 10 to the 9 tons per year. The marine organisms, another 120. and other terrestrial organisms around 53. And that's net, so that's after it's respired back out. So we're gaining about 258 billion tons of carbon annually each year. And this carbon is used to effectively power the planet. So life on Earth ultimately depends on photosynthesis, and all that carbon gives rise to our food, and all of the past fossil fuels that we use are all derived from that influx of carbon, and the exhaust gas is oxygen. And this is a slide that I show at the beginning of a 200-level lecture to warm people up. But it reminds the students that photosynthesis is all about harvesting the energy of the sun and converting it into chemical energy, and then the chemical energy is used to fix carbon dioxide at that large amount, the 258 billion tons per year. And there's an overall simple equation that you can write for the process where carbon dioxide and water are combined with light to give us sugars, and there's the oxygen being given off. But photosynthesis does more. It's not just food and fuel and oxygen. It's also building materials, heating materials. It gives rise to clothing, obviously cotton, but subsequently wool, and even plastics, because fossil fuels, of course, are the feedstocks for plastics. So it's very hard for any process on Earth to be detached from photosynthesis. And getting ready for this lecture can make you a bit nervous, but this number makes me more nervous. That is, at 3 o'clock this afternoon, the number of people on the planet. And it's increasing by about 150 a minute. And if you do the sums, that's a net gain. That's after those of us who have left. These are the net gain, is 78 million a year. And 78 million people are going to want photosynthesis to deliver these goods to maintain the standard of living that we're used to. They're going to want it as well. So one of the challenges, of course, in photosynthesis research then is to contribute to acquiring a source of energy that will help sustain the lifestyle and the quality of life for those of us who are here and all of those who are rapidly joining us. So photosynthesis is at the heart of the carbon cycle. 258 billion tons of carbon are fixed each year, and that's the net gain even after respiration, and the net gain also drives the food chain. This is a vegetarian food chain. The elephant is eating the leaves, but even vegetarians don't make it, and when you die, the microorganisms recycle you, and the CO2 comes back to the atmosphere again. And photosynthesis is really at the heart of that process. But what I'm going to do today is delve into the machinery of photosynthesis a little bit, go into the canopy, go into a plant leaf, and go into a plant cell, and look at the chloroplasts that are there. As Frank alluded, these are actually derived from cyanobacteria originally, but now they are living out their life in plant cells. And they have an external membrane and an internal membrane system. And the internal membrane system is the site where the photosystems are located and where the light energy is absorbed. And they generate two major compounds. One we will abbreviate as ATP and the other as NADPH. And those compounds, which are the result of the energy transduction from absorbing light, are used by a series of reactions that happen in the aqueous compartment up here to fix carbon into sugars. And the enzyme that does that is called ribisco. It stands for ribulose bisphosphate carboxylase oxygenase. It's a long name for an enzyme that's very abundant. It's the most abundant protein on the planet. And so this is the the overall summary of how photosynthesis works. The thylakoid membranes have the photosystems. They absorb the light energy. They generate these products, which can then be used to fix carbon into sugars by the Calvin-Benson cycle, and Calvin and Benson, of course, are the investigators that elucidated that pathway in the 1950s. So you can actually take this direct route and use it to make biofuels, and many people on the planet are trying to do this, and this will be part of the way we solve our energy problems in the future. So the pathway for biofuel production, we see light coming in, it's being absorbed, it runs an electron transport chain on those thylakoid membranes. We make NADPH and ATP, we fix carbon into carbohydrate, and then if you grow algae, here they are growing in nice plastic bags, you have the advantage that you're not competing with the land for food, and so you can use algae to generate products without competing with the food supply, which is, of course, a critical factor. And you can divert carbon metabolism towards, for example, extra lipids, and that can be turned into biodiesel, or you can convert it into substitutes for petrol. That butanol can go into an engine of a car and run a car with the current infrastructure. And in fact, our lab even has played around a little bit with making butanol. But the area I want to get into today is going to be more focused on Photosystem II. There was a mention of efficiency in the introduction there. The efficiency of getting down to this end of the pathway is actually quite low. It's probably down around 2 or 3%. And of course, organisms are actually designed to reproduce. That's what they worry about being efficient at. And so there's plenty of light, so they don't overdo the capture of light. The high efficiencies are in the initial reactions, which are close to 100%. And I think that's partly because if you're in a shaded environment, you want to capture every photon that comes, so you've got a very efficient system to capture light. And so it's, can you adopt this very highly efficient front end for biofuel production? That is one of the things that we'll talk about today. So it's hard to remember everything about photosynthesis. Last year, I was lucky enough to have a book that came out that sort of covers the whole field: plastid biology, energy conversion, and carbon assimilation. And you can see from the look on my face that I was very chuffed by that. And the Springer put this up on their website. The book will be a valuable resource for advanced students, teachers, and researchers of photosynthesis, and I hope that's the case. So it's 900 pages, and we're just going to talk about one chapter that deals with that protein there and focus down on Photosystem II. I want to go back to the beginning, and the beginning is going to be the origin of life briefly, just to put a context on Photosystem II. This is actually my beginning. I was born in January. This is, I think, April the 21st, 1957. And sadly, my mum and my dad and my brother are all gone now. But I'm still here, and though I'm looking at the camera, I cannot tell you who took the photo. And when you try and extrapolate back to the beginning of life on Earth, there isn't anybody that can tell you what happened. And I'm just going to pick my favorite scenario that has come from two researchers, Mike Russell, working at the Jet Propulsion Laboratory in Caltech, and William Martin, who's professor of botany in the University of Düsseldorf. And they have investigated hydrothermal vents off the mid-Atlantic ridge. And they're not right on the ridge. The vents that are right on the ridge are in contact with the magma, and the water that comes up is too hot. These ones that are off to the side, the water is not so hot. And down in the crust, there's a process where the high-pressure water, which there is about 150 degrees centigrade, is converting these magnesium silicates, olivine and similar ones, into serpentinite, and there's a process called serpentinization. And as a consequence of that chemistry, there's an upwelling of water that's coming up to the surface that is very alkaline, rich in hydrogen, brings up hydrogen sulfide and ammonia, and it's coming up to the surface, forming these precipitates, and there are, in these structures that are forming, there are microcells that are about a millimeter or a few micrometers in diameter. And the ocean, particularly in the Hadean, when this is a process going on right now, but this is a process that has probably been going on since the beginning of the planet. And the acidic Hadean ocean would have had 1,000 times more CO2 than we have now, which would have made a very strong acid-alkaline gradient across these microcompartments in these structures. And that offers two things. It offers a source of energy, and it offers a source of concentration for the origin of life. For this, you can see the plume coming out at the top of one of these things, and this symbolizes the microcompartments that are being formed by transition metal sulfide deposits that are forming as the warm water comes up and meets with the ocean. And what is postulated to occur is, and there's certainly today, there's methane, there's formate, there's pentane, there are organic molecules formed here, and so there's all of the thermodynamics are correct to get prebiotic chemistry that will give us precursors, amino acids and sugars and bases. They're concentrated. It allows for a nice environment for the generation of the RNA world and the formation of ribosomes. This then would advance on to the DNA era. And because you've got that pH gradient, the ATPase that makes ATP evolved in these structures as well. So this envisages that the universal ancestor for life on the planet is actually in a rock. It was not a free living cell. and then this subsequently moved on, and there was some more biochemistry invented, and these microcompartments developed membranes, the archaeobacteria being one form of prokaryote and the eubacteria being another form of prokaryote. And you can distinguish them by their many factors, but one of them is the way they make their membranes. And then these would have gone off, and we would have had Darwinian evolution. So at this point, there's no photosynthesis. But then as you move into the archean, you get cytochromes formed and quinones, electron transport molecules that expand the capacity of those cells to do electron transport and to harness proton gradients that they can now use because they form membranes and their ATPase is sitting in the membrane. And you can see from a heme to a chlorophyll that there aren't too many steps necessary to get an extra ring and a tail. And as soon as bacteria were photosynthetic, then they could move away and not be dependent on chemical nutrition, and they could actually use light to drive their systems. And evolution is really starting to gather speed. And there is about 2 billion years here of the age of the bacteria when they dominated the planet before one of those archaeobacteria engulfed the eubacterium and gave rise to the mitochondria and the eukaryotic cell. So on that pathway at around 2.5 billion years ago, things change in a big way, and we get oxygenic photosynthesis. So all of these systems, I'm just going to use this slide to just briefly reiterate the point of these proton gradients. Whether or not you're in the eubacteria, which gave rise to the mitochondria, or a photosynthetic bacteria, or in a cyanobacterial electron transport chain, all these electron transport chains are designed to move protons across the membrane, and that puts a proton gradient that can be used back out through the ATPase that was formed way back in the mounds at the hydrothermal vents. And this then allows you to generate the ATP, which is used to run metabolism in cells. And this concept was first put on the table by Peter Mitchell in a paper in 1961 in Nature, and he eventually got the Nobel Prize in Chemistry in 1978, and he called it chemiosmosis. So oxygenic photosynthesis is harnessing the power of chemiosmosis and allowing for an efficient electron transport chain. And when you see this protein for the first time, you might say, wow, that's incredibly complicated. How did that suddenly arise 2.5 billion years ago? But remember, we've already had the ribosome, which is also incredibly complicated. We've already had the ATPase, and we've already had a number of respiratory enzymes evolve that bind cytochromes and can use quinones to shuttle electron transport. So to get this complexity was clearly within the scope of nature. It was not such a huge challenge. And we'll learn as we go along that one of the things about Photosystem II that's very interesting is that it turns over very rapidly and therefore provides a model system for us to study how nature actually went about building such membrane proteins that are so complicated. Certainly the arrival of oxygen can be described as the Big Bang in biology because oxygen itself then allows for the development of large body plans and the development of large organisms. So you can refer to Photosystem II also as the engine of life by generating this oxygen. And the other remarkable thing about it is that if you want to do that reaction yourself, you would have to heat water to 2,000 degrees centigrade, whereas it decomposes at ambient temperatures in plants out in Dunedin at any time of the year. So this enzyme solves the thermodynamic problem of getting water to break down and to give us the electrons that can go down the electron transport chain and the protons that can be coupled to ATP production and also, of course, to hydrogen production. So Photosystem II, the engine of life, it's also produced oxygen that gave rise to the ozone layer, and that means that it protected us from UV radiation, and that allowed organisms also to spread out across the planet. And it is also, as I mentioned before, made possible the evolution of large body plans to support aerobic respiration. So the advent of this enzyme has a major impact on our environment. This is me with mumps in 1963. I don't remember it hurting. I certainly had chubby cheeks. But so here I am being exposed to a virus, and many of you perhaps had the same virus, and we survive without any problem. And it's because we've got some kind of immunity from prior experience, and our bodies, in this case, fight it off at about 6 days. When Photosystem II did this reaction and produced oxygen, it creates a problem because oxygen is, in a sense, is also toxic because it forms reactive oxygen species which attack proteins. So the fact that it existed and that this metal center in here became this complex here, this oxygen-evolving enzyme, and it worked, it says that there must have been some form of pre-protection to stop this from auto-destructing when it started to make oxygen. And that leads to a couple of, I guess, speculations, but one of them is that the development of electron transport chains must have actually resulted in a need to develop antioxidant machinery. And remember, there was no UV at this point, so you've got UV bombarding water, splitting water, and you're getting oxygen from that. So it looks like there was some oxygen before oxygenic photosynthesis. And the enzymes that dissipated the reactive oxygen species, and I summarized some of them here. Here's oxygen. If it picks up an electron when it shouldn't, it makes a superoxide anion, and that's very reactive. If it interacts with an excited chlorophyll, you get an energy transfer and you get singlet oxygen, and that's very reactive. You need to be able to manage these species, and it seems that there was likely enzymes such as superoxide dismutase already present because of those nascent electron transport chains. And in fact, the manganese center there is probably the origin of the manganese center that's in the oxygen evolving complex. So it probably evolved from that. And there's one other neat thing that when you stop and think about this issue, and that is if the UV is coming in and bombarding the ocean and we're getting oxygen released, in the early atmosphere, that oxygen will be snapped up by iron in the crust, just like that. And so that leaves the hydrogen, and the hydrogen's going to go wafting off into space because gravity is not strong enough to hang on to hydrogen. So at this point, 2.5 billion years ago, we're on our way to Mars and Venus. And so this enzyme putting oxygen in the atmosphere and therefore recombining with the hydrogen to keep the water on the planet is also, of course, something that we're all very thankful for. So I got interested in it and I decided I'd go and study it. And I went to Illinois, and they got there in 1981, and I was going to study the electron transfer to quinones in Photosystem II, and I was interested in the proton coupling of that and the proton transfer across the membranes associated with making ATP. That was what was on my mind when I accepted the project. And the project I was given was called the bicarbonate effect, and it takes just a moment to set the context. It goes back to Otto Warburg. Warburg was a very successful biochemist and got the Nobel Prize for his work on cytochromes in electron transport chains, and I have his biography, Merv Smith, and I will give it back to you. And Warburg was, I don't know, he was fixated on the fact that the oxygen in photosynthesis came from CO2 and not from water. And when I was 1 year old, he did an experiment here where he flushed out these algae with argon and got rid of the oxygen, got rid of the CO2 and slowed down the rate of oxygen production. And he said, hey, that's why, that's showing you that the oxygen comes from CO2 and not from water. If I remove the CO2, I slow down the oxygen. That idea goes back to the end of the 18th century. Actually, Joseph Priestley in 1771 had done a very famous experiment where he had a bell jar and he put a candle in it and he burned it and used up all the oxygen, then put a poor mouse in there and the mouse suffocated. And then he put a plant in the bell jar, as you would, and then put a mouse back in, a different mouse, and then that mouse ran around very happily. And so Priestley said, look, I've discovered oxygen. The French would say Lavoisier discovered oxygen. The British would say Priestley discovered photosynthesis. But this man, Ingenhouse, who's Dutch, helped Priestley out because Priestley couldn't repeat his experiment, and it was Ingenhouse that showed that he needed to put light on the plant to make this happen. And his contemporaries, Sennenbauer and de Saussure, also showed that you needed CO2 and water. And they came up with the idea that here is an equation that summarized Photosystem II, and you cleave the carbon dioxide, you get the oxygen, and you get the carbohydrate. And that stuck around until 1932, when this guy, Cornelius van Deel, working at the Hopkins Marine Station in Stanford, working with purple sulfur bacteria, so bacteria that don't evolve oxygen, but actually use hydrogen sulfide, and he could smell the sulfur, and he proposed this important equation where he said, right, the electron donor is hydrogen sulfide, I get sulfur. If I generalize that, then I could say it's H2A, and I'll get 2A, and if it's a green plant, that's water, and I'll get oxygen. So he proposed correctly in 1932 that it was oxygen. And that was proved by two key experiments, one in 1939 by Robin Hill in Cambridge, who isolated his a chloroplast from algae, extracted the thylakoid membranes, got rid of all the machinery involving rubisco and CO2 fixation, and shone light on those thylakoids with an electron acceptor present and measured oxygen coming off. So that was, yes, Van Niel is correct. And then Martin Kamen and San Ruben, working in Berkeley, got themselves a collection of 18O water from the Lorentz synchrotron, and they did an experiment with mass spectrometry to show that the oxygen that was evolved from this water was always the 18 isotope and not the 16 oxygen isotope that was in the CO2. So it looked like it was a done deal. But then someone in Gavinji's lab rediscovered this idea of Warburg in 1958, and they did an experiment where they were able to scrub out all of the CO2, and they stopped the reaction of oxygen evolution, which is by this time was called the Hill reaction after Robin Hill. And so they could recover that by adding bicarbonate back. And then I came on the scene, and this is actually at Gove's retirement in 1999. And I decided I was going to look at this bicarbonate effect because there was evidence from this lab that it was on the quinone reactions and not on the oxygen evolving reactions. And to do that, as a botanist, I had to build this. And this took me 2 years, and it was a lot of fun, and I learned a lot of things, and I wrote it in a computer language called Forth, which I don't think anybody else knows anything about. I've never heard of it since. But I bought a book, I read it, and programmed this thing, and I built this machine. If I take the lid off that, I had to hand wire literally hundreds and hundreds of connections between the pins on the chips. And there's a 12-volt power supply and a 5-volt power supply. And among all the hundreds of wires that I connected, when I turned on the switch, there was a disappointing pause that nothing happened, and then flames leapt from the computer. Fortunately, my board that I'd wired up survived, and Gavinji allowed me to buy a new motherboard for the computer. And eventually, we got the machine going. And we were able to do experiments that measured electron transport from the reaction center of Photosystem II to reduce these quinones. And so the idea of the experiment is very simple. You give a bright flash of light, and then the chlorophyll molecule absorbs that, and it puts an electron onto the first of these quinone acceptors. There are 2 of them there. And then the oxygen evolving complex gives an electron to this so that it becomes neutral again. And then the electron from QA goes all the way over to QB, the second quinone acceptor. And then you can repeat the cycle, and the reduced QB, I've lost my cursor there, diffuses off down the membrane to a place where it can be oxidized and pass the electrons along the chain. And if you actually give one flash and then wait a long time before you give a second flash, the electrons got to QB, and the fluorescence that you get back is low. But if you give a flash and then another one very quickly, the system still has the electron on QA, and it can't do the charge separation there, and the energy comes back and the fluorescence is high. So by firing off this flash lamp at different time durations, it was possible to get the kinetics of that reaction. And I show it here. This is out of my thesis, I think, this picture. Inhibited by a herbicide, DCMU, atrazine does the same thing that blocks this binding site, but bicarbonate did the same thing. And I was able to conclude in the thesis that bicarbonate was actually regulating electron transfer between QA and QB, and this is what Warburg had seen. And we also, at that point, knew that there was a non-heme ion in the system, and we hypothesized that the bicarbonate was a ligand to that non-heme ion. Unfortunately, my colleagues were skeptics, and they said, first of all, why worry about Warburg's ideas? They're nuts. People already know it. Oxygen comes from water. And secondly, they said you've got to treat this with low pH and formate to get rid of the bicarbonate, and then you have to use 10 millimolar bicarbonate to get it back. And it's reproducible, but basically, Julian, it's an artifact. So I was very pleased. In the structure that came out in 2010, sitting right next to the non-heme ion, is indeed bicarbonate. And there is a channel that goes out to the surface to show where the bicarbonate can diffuse through. And here's the QA and QB electron carriers that are there in the structure. And this is a modification that is different from the anoxygenic photosynthetic bacteria who use a glutamate residue there. So one of the questions, of course, is why do higher plants use bicarbonate and not glutamate? I'll speculate on that at the very end. All right, so I'd done some biophysics there. I needed to learn some new things. I went off as a postdoc, and I went to Japan, worked with Norio Murata, learned how to isolate proteins, hydrophilic proteins, and I had a mustache when I worked in the lab and a nice sushi jacket. And then I went and worked for this guy, Wim Vermaas, who is about 10 years younger than me. You would not know it. And he's very bright and he's burned out fast, but or at least his body's burned out fast. His brain is very sharp. So he taught me molecular genetics, and I picked up the cyanobacterial system and became the closet microbiologist. And there's Gove sitting next to Krista Critchley, who was a postdoc when I was in the lab. This is in 2000, and Krista was professor of botany in Brisbane. And finally, I wound up with Geoffrey Hine, and with Geoffrey, I learned to isolate membrane proteins from Photosystem II. And Geoffrey actually is worth a quick mention because he was the first person to prove that Mitchell was right with chemiosmosis. He did an experiment at Johns Hopkins with his mentor, Andre Jagendorf, to show that if you shone the light on thylakoids, you could measure the proton uptake just as predicted by Mitchell. And then he did a brilliant experiment where he actually incubated the thylakoid membrane so that he put an artificial proton gradient between the lumen and the outside of the prep, put it in the dark, no electron transport, just a proton gradient, added ATP and inorganic phosphate, and got ATP. And they published a paper in 63 and a follow-up in 65. So photosynthesis went with chemiosmosis early. The mitochondrial people took another decade, which is why Mitchell didn't get his Nobel Prize to 78. So be it. I came to Dunedin. This is the lab as I saw it when I came in April 1994. It was empty. And this was the door to Merv's office and also the door to George Peterson's office when George Peterson was there. And I think this lab was actually built for George to nip out of the office and do science when perhaps there was more time for HODs to do science in those days. Merv was very good to me. I think he gave me a budget of $10,000 or $20,000, and we gradually built up the lab. And there's the first crew of honours students starting in February 1995. And Nick Fleming now is working over in pathology. Victoria there is a lecturer at Lincoln. And Shannon is, I think, a senior scientist now with ag research. So they've all gone on to do science. I stayed on London Street. I guess Merv arranged it for me to have a bed and breakfast for a little while. And then I had to find a place to live. After all those years postdocing and being a PhD student, I didn't have any money. And so I could walk, so I walked around. Somebody said, you know, Mary Hill's not that bad. She's not far from the university. I found this little place being rented on the first weekend in town. I rented this room. It had nothing in it. And you've got a job, right? You have to think, what am I going to do? You actually need clean clothes. This is actually the first problem, right? I couldn't find a laundromat in this town, at least not in walking distance. So I was off to Arthur Barnett. I think I paid cash for the iron and the ironing board, but the washing machine and dryer were on credit. After the end of the week, sleeping on the floor in a sleeping bag, the next thing I wanted was a bed. And so I went and bought a bed and a blanket to keep warm and a duvet, and so I've still got the bed. This is my first lecture. I've been here about 6 weeks. Kevin, who's out there in the audience, with Paula Jameson, had established this wonderful course in plant biology, looking at molecular and physiological plant biology. So it was a joint effort from the very beginning between botany and biochemistry. And the class was always very small. Diane there went on and did, I think, a PhD with Paul Guy. Jenny did one with Dave Burrett and Peter Bannister. Tom went off to Australia. This guy, Barnaby May, was an honours student in chemistry, and he got his PhD, I think, in Canterbury. And there's Shannon, and she came on and joined my lab. So they pretty much all went on in science. Over the years, we changed the name to Plant Biotechnology, and then we've sort of revamped the course. We now call this course Applied Plant Science, and we focus on real practical issues of where plants are actually making a difference to the planet. So David Burrett does cleaning up pollution and Chernobyl with plants, and Tina Summerfield does biofuels, and Richard McKnight talks about the importance of secondary plant products for commercial exploitation. And the students like it, and we had 37 students this year. And I think, you know, it's an important thing for our university that we have a strong plant biotechnology thing, because New Zealand needs plant biotechnologists. And it's nuts that we don't make more of it, you know. And, so we've got to keep them coming. Yeah. While that was happening, back in 1995, there was a floor being built, and this is me standing in my office there, and that's the door back there, and there's Kevin standing in his office too, so he also would creep up there, and that was where he was, and Richard McKnight's now in that office there, and Lynette is in this office here. Lynette joined us most recently. And I'd go up there almost every night, and I'm going to keep you here for a long time, I have to be careful, but I would go up there almost every night and sort of think about the lab and how it was working, and often you'd hear the footsteps coming down the corridor, and it would be Merv having a look to see how it was going on. And this is, I think he didn't even know I was there half the time, but this was, I think a great achievement for Merv that he got that top floor on biochemistry for us, and the lab's running very well. Here it is 10 years later with how we went off to work on biofuels in Denmark with my dad at 86 coming out to see me in 2006. So the lab is going strong, and we all enjoy working there. So what do we do there now? As I said, I was interested in the difference between photosynthetic bacteria and the green plant transition between these photosystems. And the reaction center associated with the photosynthetic bacteria is depicted here, and it was a milestone in biochemistry. It was the first membrane protein ever crystallized by Mikkel, Deisenhofer, and Huber. They were the scientists that did it in Germany. And they, of course, got the Nobel Prize in 1988 for that achievement. But if you compare that with Photosystem II, you can see that there's a great jump in complexity from this bacterial reaction center to Photosystem II, and the difference is that this one can split water. So the question then becomes, why this additional complexity? What is the necessity for all of this complexity to allow it to split water? And we've sort of been looking at that as we've moved on, particularly in the last 5 or 6 years. And there's ways that you can do this. These are cyanobacteria growing in flasks, and it's very easy to knock out a gene and look for the effect of that. And the proteins inside this dotted line, these membrane-spanning helices looking down at the top of the complex, actually they all have their counterpart in photosystem I. There are 2 photosystems in electron transport in plants. And they also have their counterpart in different kinds of bacterial photosynthetic reaction centers. The unique proteins are the gray ones that are all around the outside like a horseshoe. And there's some that sit at the monomer-monomer interface. And so we've been sort of knocking out the genes for these and looking to see if there was any phenotype. And for example, when we knock out that one there, we stop the assembly of the complex. And we've also studied a number of these proteins that sit as a cap over the oxygen-evolving complex. And they provide a channel for the water to get in and the oxygen to get out and the protons to get out. And we've also looked to see what happens when you remove those. This work got us interested in biogenesis of the complex. And what I've attempted to do in this little diagram here is to summarize the photosystem II with that core set of proteins that are common to many reaction centers, and then put around the outside all of the proteins that are unique, and they're given letters of the alphabet, and each one is relatively small. And then those are the 3 proteins which you'd see down here, and there's the manganese center, which is sitting there. So that's the crystallographic system. And now it turns out that when you purify photosystem II, there are a whole bunch of non-crystallographic proteins associated with it as well, and there's 5 listed here, and it's a list that's growing all the time. And there are 3 others which seem to play a role, and these proteins are not in the crystal structure, and yet they're required to get to this system. So they're part of the photosystem, but they're not there in the mature complex. And so we were interested in finding out what do they do and how do they work. And you can see the attraction. Remember, this complex formed once all that time ago, 2.5 or more billion years ago. So it's possible that by exploiting this system to study the assembly of this complex, you can learn something about how very ancient protein complexes were assembled at this early time in life on the planet. These 3 proteins are particularly special as they have a lipid attachment with them that allows them to bind up to the complex, or at least that could be the explanation for it. And we actually embarked on an effort over a period of about 4 years to get the structures of these 3 proteins, and we were successful. And I'll just show you, initially we started out looking at cyano-Q, and I'm sorry about the names of all these things and all the letters, but that protein, we knocked out the gene for it, and in fact, we did it in a way where we made double mutants. So we knocked out the gene for that, and the gene for that, and that was one mutant. We knocked out the gene for that, and the gene for that, and that was another mutant. And likewise, the gene for that, and the gene for that, and that was another mutant. And then we compared growth. And the work was done by Tina Summerfield and Jackie Daniels, as she now is, and I expect Jackie is here somewhere. And Jackie's been the technician in the lab since 1996, and so she's just, so it's sort of keeps me on the straight and narrow. And Fiona became a PhD student. She's now in Colorado, still working in science, in biofuels. They all like to go to biofuels. So what we found out was this particular double mutant didn't grow, and it was the first evidence that that Q protein was PS2 specific, but if you gave it sugar, and so it didn't need photosystem II, it could grow fine. So we were on to the fact that that was a PS2 specific protein. And then we went ahead and got the crystal structure, and Simon Jackson did it as part of his PhD, and Rob Fagerland, who joined me as a postdoc after getting his degree with Sigurd, and he's now in Penn State, and that's presumably his first Halloween coming up here. And he helped us, and we got this structure, and now we're able to study how the protein-protein interactions between Q and the photosystem II complex by looking at the structure and looking at comparative sequences. And in fact, a student came to me yesterday and asked if they could do just that, and I said, yes, you can. We also looked at this 27 protein. This is a lovely example where there was a collaboration in the department to solve a problem. Sigurd co-supervised Peter with me, and Sigurd is the crystallographer, of course, and we tried quite hard with crystallography, and we had Catherine on the committee, and Catherine likes to work with NMR to solve proteins, and so she was able to say, well, all right, I can put you in touch with my colleague Mark Hines over at the WeHi, and we can do this one by NMR. And so we did, and Catherine helped Peter learn how to do the assignments, and we solved that protein. And both those proteins were first. They were competitive, and we got there first. Mark Hines also helped Simon get a third one, the cyano-P, and we were second there, but Simon's structure is better than the other one, okay? It really is. I'm not just saying that. So that's great. And so we solved those three proteins, and now we're busy thinking about how they will interact with the photosystem. And this one's the most enigmatic, and you can model, and the slide is being chopped around a little bit, but we can still see what it says. You can model how that protein can bind to photosystem II, and it binds in the lumen when you model it in the computer. My students hate the fact that I like to make models. They say, Julian, it's going to be wrong, but I just want to tell them all models in science are wrong right up to the last one. If you don't make a model, you can't test it. So here we are. Here is a wrong model for all of my students that we can test and refine, but it seems to be associated with the luminal side of photosystem II. And it seems to be associated with a cofactor that is involved with the assembly of the oxygen-evolving complex in a process that's known as photoactivation. And I just summarized that in this diagram here. You start with the enzyme in the dark, and there's no manganese bound. And if you put a photon in, you can oxidize one of these manganese, and it sticks. And then if there's calcium present, it moves to the next state, and you can see there's a calcium in the final assembled complex. And then you have to put in another photon to lock in that next manganese, and then there's a dark rearrangement. And if you give a few more flashes of light, you arrive at this system. And it looks like the one possibility is that cyano-P can contribute to that process, to actually make the oxygen-evolving complex ready for activity. And plants will be doing this at dawn as the sun comes up. Then the next thing that you want to worry about with photosystem II is actually how does this water-splitting reaction occur? And so you make, this would be the complex that's just been made by photo assembly, and then it's got to perform the chemistry. The chemistry it's got to do is actually interface oxidizing two waters, which is a 4-electron chemistry, to the 1-electron steps associated with the photochemistry. And Pierre Joliot, who's now 80, I've got his picture over here, recognized in 1969 that there was a periodicity of 4. If he gave 4 photons, he needed 4 turnovers to get oxygen. And then Bessel-Koch, who unfortunately died in 1979, in 1970, Bessel-Koch came up with a model that said you need 5 oxidation states to achieve that, and he called them the S states, S0, 1, 2, 3, 4. And it's a testament to the insight of these guys and the great experiments they did that is still the paradigm to study this reaction in 2013, even though it was hypothesized in 1970. And the filling in of the oxidation states reflects the progress made in spectroscopy between then and now. I just point up here, that's Madame Curie and Pierre Curie. And remember, they got the Nobel Prize in Physics for discovering radioactivity, and then unfortunately he was hit by a horse and cart. But she went on and discovered radium and polonium and got the Nobel Prize in Chemistry. And that's Irene Joliot-Curie, she married Frederick Joliot, and she got the Nobel Prize for artificial radioactivity, and she is his mum, and unfortunately he didn't get the Nobel Prize, but he did give us this, and it really is a major contribution. So I can just briefly show you how it works. I called it the ticking clock, and the clock is ticking, and I imagine some of you are saying, Julian, okay, that's good, we're moving along now. But I'll just do one of these for you. What I've done is I've stripped away the protein environment so you can see the cofactors. And this is the stuff that's got the high efficiency. This is the stuff that has a quantum yield of about 1. And so when a photon comes in, it will advance this system. First of all, the photon goes to the chlorophylls that are at the reaction center, which we call P680, and it'll move the electron up to the quinone, and there's the iron where the bicarbonate binds, and you'll get that electron transfer reaction, and then this residue here will replace the electron that's gone, and then the manganese in this complex will replace the electron that's left that, and you'll see that this manganese 3 steps up to an oxidation state of manganese 4. And this repeats for each of the flashes until you get to the final state that nobody's ever got any information because it's so transient. And that's then decomposes, oxygen comes off, and the whole thing resets, and you bind 2 more waters, and you can do the cycle again. The structure, we now have a structure of the manganese complex. It's a manganese-calcium sort of distorted cubane complex, and it's got 4 waters bound to it, frustratingly, in the crystal structure. And it was believed we were looking at that state when the structure came out in 2010. But it's now clear that the amount of radiation that's been used to get this structure is probably reducing the manganese back, and it's now unclear if we're in this state or even below that oxidation state. So it means that it's very close to a real model and resolution now, and then people will be able to do the biomimetic systems based upon it. But got to overcome this radiation damage problem. I would say in just a couple of years now, we will have the full mechanism of this reaction. So that's the promise of getting that catalytic site. We also have been interested in the protein framework that surrounds the system, and I've just got one little story here that just shows you the complexity of the photosystem at the level of proteins. And you've got to make the system, and that's a membrane sitting there. And initially, there's a complex that forms between one of these transmembrane spanning proteins that were gray in one of the previous diagrams, and a part of the reaction center, which all of these names are dreadful, which we call D2. And this then has to combine with another complex, which has D1. And of course, you'll see this has come in with 1, 2, 3, 4, 5 polypeptides. So you still need to actually understand how that complex was made before you get to this pre-complex. Certainly, this protein here is to do with folding of this protein as it comes in the membrane. It's got 5 membrane-spanning helices. It's got to bind chlorophyll and all of those cofactors. This protein probably brings manganese. And this comes as a precursor, and it has to be processed. And the processing of that involves another polypeptide called YCF48, and we make that one blue because we're actually actively studying that one in the lab right now and how it interacts in the biogenesis pathway. You've got to clear that away, and you get a mature form, and then you need to bring in a protein complex which is bringing more chlorophylls, which will operate as an antenna that can pass the light energy to the reaction center that's sitting in here. And again, the YXHLMT make it through to the crystal structure, but the 28 and this SLLO933 do not, and we're interested in the role of that one, so that's another protein structure that's available, and you can study how it enables this antenna to come together. Then you've got to bring in the other half of the antenna system, which will fly in, I think, from this side any moment. And our PSB27 protein that Peter solved is interacting with that complex, and we want to understand the protein-protein interactions there that allow that to come together. Then you've got to get that out of the way. Then you've got to do the photoassembly of the actual oxygen-evolving complex, and then you've got to put on the extrinsic proteins, and we hypothesizing our Q comes in at this point, and then this thing has to become a dimer. Then it's working. And we can disrupt this interface by attacking, making mutants there, and we've done that too, and it still works, even if it's monomers. But it's working at the edge of sort of chemistry. There's the very high redox potentials. The redox potential, that's its desire to take an electron from water. It is the highest oxidizing component in biology, and it actually damages itself, so you get a damaged reaction center protein. And then the system, in a coordinated way, has to go to a monomer, has to remove those extrinsic proteins. It's got to get the CP43 antenna out of the way so proteases can get in there, get rid of the guy that's damaged. Ribosomes have got to make a new protein, got to insert that in there. It's got to be processed again. The YCF48's got to come back in, and then the CP43 has to rejoin the complex. And we actually think that the movement of CP43 in and out from the reaction center may have something to do with why the system has switched to using bicarbonate in the reaction center and not the glutamate, that actually there's part of the signal comes, bicarbonate's involved with part of the signal that controls that little shunt there. Okay, we've got to get rid of the 27, we've got to photoactivate it, and we've got to put the cap on, and then it's got to become a dimer again. And the important thing about this is that this complex is turning over all the time. It's repairing itself so it can sustain oxygen evolution. So it's got intrinsically built into it self-repair. So by understanding the self-repair that's going on here, then you can design that and build it into biomimetic systems so that you can prolong the lifetime of these devices so that you can have artificial water-splitting complexes that will give you hydrogen as fuel. So to get to this, you want to understand the self-repair systems, and that's what we're trying to do when we look at the assembly of the oxygen-evolving complex or the assembly of this complex. And then you can ask how that can be contributed into the design of biomimetic devices that will perform water splitting. And there's another critical point, and it's very critical for the 78 million people that are about to join us, is that if that repair process, if it gets out of kilter, so when you're in a stressed environment, the rate of damage doesn't keep up with the rate of repair, and therefore crop yields go down. And so if you can study that repair process, it was quite complicated, but by studying it in plants that are resistant in harsh environments, then you identify traits that you can transfer over into crop plants so that you can then grow the crop plants in marginal lands to feed people. And as I say, there's 78 million more in 2014 than there is in 2013, and it's not stopping yet. One last point, and this is the synthetic approach. This has come out of the chemistry department at Penn State and the aptly named Center for Bio-Inspired Catalysis for Solar Fuel Generation at Arizona State University. What these guys have done, you can read it there. It's electron transfer reactions in the media-based dye-sensitized titanium oxide photoanodes showing molecular structures of the bit mediator 3p rubidium dye and the SEPA linking group. So what have they got? They've got a dye here, a ruthenium dye, coupled to a titanium oxide electrode surface, and then they've got this carboxyphosphonic acid linker to the iridium oxide here, which is mimicking the oxygen-evolving complex. So this is a nanoparticle that can split water, and then they've got this the benzimidazol phenol, there, that guy there, as the mediator to return the electron to the ruthenium dye. So they can make this thing go, but look at it's only 2%. Okay, for these guys, I don't want to belittle that. That's a big deal. That's a totally synthetic, photosynthesis in principle based reaction center that's splitting water. But it says it's possible and we can do it, but one of the problems is that ruthenium and iridium are some of the most rare elements, transition metals. And so you can't really expect everyone to have one of these on their roof. You've got to understand the plant system which uses manganese and calcium, and that's abundant and no problem, and then take that and then build that system and build it in, and that's the goal. So when I finish up with my 200 level lectures, possibly the 100 level lecture, so I get them mixed up, I finish by saying, putting up this slide, and I'd say that understanding how Photosystem II solves the challenging thermodynamics of water splitting will facilitate the design of biomimetic systems to fulfill the vision of Jules Verne. And Jules Verne actually said this at the end of the 19th century. He said, "I believe that water will one day be employed as fuel, that hydrogen and oxygen which constitute it, used singly or together, will furnish an inexhaustible source of heat and light of an intensity of which coal is not capable. I believe then that when the deposits of coal are exhausted, we shall heat and warm ourselves with water. Water will be the coal of the future." Thanks.
So it's my job to thank Julian for an outstanding lecture, and it really was an outstanding lecture. And I think behind every outstanding scientist there is an incredibly neat puzzle, and you've got an incredibly neat puzzle that is your passion, and that really shows through. And so this was just an amazing tour de force. And I think we started out really with something we know, right? We all know how important the sun is, how much energy the sun provides the Earth, and how that energy has the capability of revolutionizing our whole civilization. So we came here knowing that, but I think, Julian, you delineated for us how much more we know in terms of, I mean, we know the names of all the proteins involved and all the lipids and all the metals, but beyond that, we actually know the detailed molecular structure of how the system is laid out, which stresses how important molecular structure research is in biochemistry. So because we know the detailed molecular structure, we are actually beginning to understand how the electrons flow and how the photons flow and how the protons flow, which is really absolutely phenomenal, right? And to me, what that means is that, so the next promise of biochemistry is the ability to go in and sort of tinker with the system, right? So you can tinker with the system to improve crop yields. You can tinker with the system to get the biofuels that we need and the hydrogen that we need and the butanol that we need. And so that's going to be happening over time now. And so we're going to be looking forward from new insights from you and new insights from your students. And starting out with the 78 million people that are going to be added on this year, we're going to need you to succeed, so we're counting on you to succeed. So that's just, and we expected. And so I want to end up really by thanking you. So thank you for that really, really neat lecture. And I also want to thank you for the years of service that you've given to the department in terms of not only the outstanding research, but all the teaching and service. And on a personal level, I want to thank you for serving as my deputy HOD for the last 4 years and for being a colleague and a good friend over the last 8 years. And we have a small token from the department and the school and the division and the university that we'd like to present you with. So if you'd come up. Thank you very, very much.